He'll be telling us about challenges of isoform identification and discovery using long-read sequencing. And for our main speaker today, we have Dr. Raymond Ng, a professor at the Department of Computer Science at UBC, and his talk is titled Deep Learning for Biomedical and Bioinformatics Research. And our event today is sponsored by the following, so thanks to our sponsors. And we are also webcasting our event today. And this is our team, the Vanguard team, so thanks to everyone for organizing and setting up the event today. And please follow us on Twitter to check for any updates and any events for our future talks and seminars. And yeah, like I said, the event is being recorded through webcast. All right, so our first speaker today is Ravi. So Bara, would you like to come up?
Hi everybody. Thanks for coming. So my name is Bara Rabi, and I'm a student at the Hatch Lab at UBC. And today I'm going to be talking to you about my work on the challenges of isoform identification and discovery using long-read sequencing. So a bit about what do we mean by identification versus discovery. So typically, when we say identification, what we mean is that we have a pre-annotated set of gene isoforms, say from a database, something like Ensemble or GeneCode, and you do your sequencing experiment, and then from the reads that you have, you try to annotate these reads with these pre-known set of isoforms. And typically, you end up with some subset of the reads that you cannot fully explain with the database of known isoforms that you have. In discovery mode, what you're trying to think about is that I have the reads themselves, and what I'm trying to do is that I want to try to find from the reads how can I cluster them into isoforms that best explain them, considering the sequencing errors, the variation that could occur at the different sequencing steps. And you could end up with different results when you're in identification mode versus discovery mode, and you would go for discovery for different purposes, which could be maybe important when you're doing something that has to do with cancer and you're looking for novel isoforms that maybe that could be cancer causing. So there isn't a question of how important is discovery, and another way of looking at the same question is to say, well, how much are we missing by only doing identification and only relying on the database, something like GeneCode. So these are two numbers that kind of illustrate the problem that we have at hand here. So there's this 2016 paper which they've compiled about 20,000 RNA-seq libraries that are short-read sequencing libraries, and they've managed to identify over 3 million novel isoform junctions that are well supported by some of these libraries. I think they had a threshold of 20 or 30 reads to support each novel isoform junction. And then there is this other paper which is much more recent. This one, they used long-read sequencing. It was a single sample, a very well studied sample, the NA1278 sample. They've had reasonably high coverage sequencing, and they've managed to find about 34,000 reasonably well-supported isoforms from this single sample that do not exist in the GeneCode database. Of course, there's a question of like how much of these are truly there versus just sequencing artifacts. But what it tells us is that there is probably a lot there that we have not still cataloged well in the database. So isoform identification and discovery using long-read sequencing is much more powerful than short-read sequencing. As we saw in the two numbers, with a single sample, you managed to extract all of this compared to 20,000 different libraries. But it's not trivial. It's not trivial because there's much higher sequencing error rates in long reads compared to short reads. And there are sequencing structural artifacts that you would start seeing in long reads that you don't see in short reads. So, for example, when it comes to Oxford Nanopore sequencing, the error rate is hovering about 15%, and it's dominated by indels. And these indels could be severe enough that you would miss small exons, and they would also cause you to have low resolution in identifying the splice site exactly. So, and there are other structural artifacts when it comes to long-read sequencing. This is one of them, for example, that is caused at the sequence level signal processing, where one molecule is split into two reads instead of being reported as a single read. And there are other bunch of them that are really annoying, like palindromic reads, reads that are degraded. There's this phenomenon with sequence-specific truncation that happens when you're doing cDNA, but doesn't happen when you're doing direct RNA. So there are all of these different issues that faces us when we're doing isoform identification or discovery using long reads. So any successful isoform tool needs to consider these artifacts and these challenges when in its design. So our work is to build an annotation-free isoform discovery tool using long-read sequencing. And I'm going to have a very quick overview of the pipeline and focus on the last step of the pipeline that we have development, which is the actual discovery of the isoforms and the clustering of the reads into these isoforms. So up to this point in the pipeline, what we do is that we have read clustering, which is just simple mapping of the reads onto the genome, trying to identify poly-A tails, to identify reads that have been split versus reads that are that have started from the end of the transcript truly. And we do our own very highly sensitive splice alignment of the reads to the gene, and we use a sequence to graph model to do this alignment. And then what we end up with is something that looks like this, where we have this aggregation of all of the alignments of the reads. And we don't exactly use coverage, but we use the splicing in and the splicing out signal of the reads to segment the gene into this structure. And we call these canonical segments of the gene. Canonical because any isoform could be explained as a combination, as a linear combination of these segments on the gene. And then we represent all of our reads in this matrix, which is a binary matrix. So each read either has a segment or doesn't have a segment. So this simplifies all of our work for the subsequent step of actually clustering them. And for our model, we use an integer linear programming model that considers two main problems, two different aspects of the problem that we have here. The first one is that we don't know a priori the isoforms that we have, so we don't know the number of isoforms as well. So we don't know the number of clusters we want to group our reads into. And the other issue is that we also don't want to overfit. Like you could imagine a case where you go to the extreme and every read is its own isoform, because we need to allow for some variation for sequencing errors. So we came up with this model that unifies these two aspects of the problem. And I'm going to try to explain it in very plainly and like the overview of what are we trying to achieve with this ILP model. So the rules of the game is that we are building this setup where you have the isoform bin and the recycling bin, and you have your reads. Any read that goes to the isoform bin needs to consider all of the other reads that also ended up in the isoform bin. We build a consensus out of those reads, and then we penalize each read by how much variation it has from this consensus. And then there's another bin, which is recycling bin. Reads in that recycling bin basically don't need to consider anything else in the recycling bin. There's a constant fixed cost for read being in the recycling bin. And then we use ILP to find the optimal solution in here. And in this case, it would be something like this, where you see this slightly brown reads is going to be penalized by 1, and then all the reads that are in the recycling bin are penalized by 2 each. What we do now is that we found our first isoform, take it out, remove it from the set of reads, and then repeat the cycle again to find the next isoform that you have. And then you repeat this until you exhaust all the reads that you have. So this is the basic idea of what are we trying to do with our ILP here. And this is an ongoing work. We're trying to still fine-tune the segmentation and clustering parameters and testing using our already built simulation pipeline and real data set that we have. So thank you very much. And I would like to thank, of course, my lab and our collaborators. And thank you very much. Any questions? Please let's welcome Dr. Raymond for his main talk.
So I'll just give a brief background on Dr. Ng before he starts. So Dr. Ng is the director of the UBC Data Science Institute and the Canada Research Chair on Data Science and Analytics. Raymond's main research area for the past 2 decades is on data mining with a specific focus on health informatics and text mining. He has published over 200 peer-reviewed publications on data clustering, outlier detection, health informatics, and text mining. He's also the recipient of 2 Best Paper Awards from the top database and data mining conferences in the world. And for the past decade, he has also co-led several large-scale genomic projects funded by Genome Canada and other major agencies in our province. And he's also, since the inception of the Proof Center for Excellence, which focuses on biomarker development for end-stage organ failure, he has held the position of the Chief Informatics Officer of the Center. And his talk today is titled Neural Network Learning for Bioinformatics, for Biomedical and Bioinformatics Research.
Well, thank you.
Because it's pointing at the speaker.
I think you'll pick up this one. So thank you for inviting me. Last time I gave a presentation in this room. I think was about 2 years ago, but then as I was walking to here, I thought, oh, last time I gave a talk to Vancouver must be like 15 years ago. That was a long time ago. I have honestly have forgotten that I would belong to the Vancouver community. Over the years, so it was great that I got an invitation, and then I thought, well, what could I be talking about, because I haven't been really doing a lot of research that I would consider to be bioinformatics. So I said, well, okay, how about if I'm going to talk about deep learning or neural network based learning, and I'm sure there's a lot of interest in this community and as well as many other communities. So I will be saying a few things. Actually, I have fewer slides than the previous speaker, so it's not going to be a very, very detailed talk. I want to spend the first half talking just about some general things about neural network, talk about some general things about deep learning, and so and then maybe we could even have a discussion at that time. Then the second half of my talk is that after we have some background introductory material on deep learning or deep networks, I want to just use one case, and in fact this is very recent research that one of my PhD students have done, and it's a deep learning framework for solving a bioinformatics problem. Now, the reason why I don't know whether you noticed that I made a subtle change to the title of my talk. So the ad that I sent, or the abstract that I sent, I was saying that this is deep learning. But then as I was looking at my slides, I said, oh, well, I think the deep network that I'm going to talk about in the second half is actually very shallow. So I don't want to get into the controversial position telling you that what I'm doing or what I'm showing you in the second half is a prototypical deep network. So I said, well, neural network, I'm sure there's no controversy. So I have make a very subtle change to the title. So let's just go. I mean, as I was talking about 15 years ago, but let's actually go back almost like 70 years ago. And in fact, this was really this was actually the beginning of what was now very, very popular, which was neural networks. So the motivation of trying to come up with a platform or a tool, if you will, or a conceptual framework, if you will, is to consider the fact that, well, if we're going to build a predictive model, and, well, maybe what we could try to do is to try to emulate what a human brain can do, what a human brain might be doing when a human brain is trying to make a prediction into the future. In particular, neuroscience has revealed that even at that time, that there's existence of massively connected networks of neurons. And so the idea that was proposed in 1943, I'm sure that was before most of us were born, was just to have a very simple mathematical model where the units consist of is trying to really simulate or emulate neurons, and then we try to connect them. And so this is a very basic structure of a network unit. And maybe I'll walk over here. It really consists of three parts. You have inputs. So on the left side, you have input links that feeds into this particular unit, AJ, different unit. And then the weights, so you have weights that you could attach to all the input links. And so that accounts for the left side of the neural net. Then on the right-hand side, you have output. It's going to do some work, and it's going to output, and then you have output links. And it's going to feed into other units, and perhaps with weights. Now in the middle there's some magic where we actually have what is called an activation function. So the input is not going to cause any change to the output when it's below the threshold of the activation function. But then once it gets activated, then you have some function G which is a mathematical function. There's obviously very, very different properties of the function G. But it basically take all the inputs, take in the weights of the input, produce the output, and then send it up. So it's a very simple, very basic, unit. Now as I said, the magic is all lies into G. You know what G is, what the function G is, the activation threshold, and then there's all these things about weights that needs to be set. So that is the basic structure of a neural network. So let me give you an example. I don't know whether you could see. Maybe it's a little bit small from the back. So this is an example that is, let's say, for segmentation. In this particular example, suppose a retail company is trying to segment its customers into 3 groups: segment 1, 2, and 3. So that's the output. And the company is going to use, on the left side, the input, 6 different inputs. One is demographic information. Second one, socioeconomic information. Third, geographical location. Fourth, purchasing behavior information. Fifth, consumption behavior information. And then the last one, attitude to product. So let's say that those are inputs. And it gets fed into this neural net. And then it's just going to compute, it's just going to have some magic, and it's going to then to say, this is the group, this is a cluster of customers that belong to segment 1, another cluster belong to segment 2, segment 3, etc. So I'm showing you an example saying that, particularly in a completely different domain, not in bioinformatics, not in biomedical, this is an example taken many decades ago, because neural network was applying to marketing and so on. Now, what this network is showing us is a few things. That all the nodes, all the inputs go in one direction from your left to the right hand side. So it goes forward. So this is called a feed-forward network, and there's no loop. It doesn't go back. It doesn't feed back into some of the previous layers of nodes. And so this is a very simple feed-forward network. Now you could think of a network then when the edges kind of loop back, like having feedback cycle. Then you have what is called a recurrent network. And I'm sure some of you, many of you have heard of the concept of recurrent network. But in a way, there's really no specific magic. Simply saying that when you connect the output, if all the nodes, all the lanes are in one single direction, then it's a feed-forward network, and then there's a recurrent network if there is some kind of loop that actually happens in the neural network. Any questions so far? So we have a simple. So the previous slides talked about in each node a very simple. Basic structure within each node, and now in this case we see a network of 13 nodes. Now this is one slide. There's a one-slide introduction of what is a deep network. So in our example, in our previous example, the left layer is the input layer. The rightmost layer is the output layer. There's a middle layer. And the middle layer is neither the input nor the output. And people generally would call that a hidden layer. The purpose of having a hidden layer is allow the user to model quantities that are not directly measured. In the previous example, if I could go back, all the inputs are directly measured. Demographic information, socioeconomic information, those are measurable quantities. And then it got fed into nodes in the middle layer in some way that we connect them using some magic function G, G1, G2, G3, etc. And so in a way, what it's really trying to do with all these nodes in the connection in the hidden layers is, so the terminology, nowadays you hear people talk about is, well, it's just engineering some features. The hidden layer is aggregating values from nodes from the previous layers, and it's creating some features. So this is why, and some features are being learned, and some features are being engineered. So you hear, people talk about how deep learning is very useful for feature generation, and that is, so the magic is simply because you have nodes in the hidden layer, and then you could wire them and to try to help you to build features. Now so all the things that I have said so far is just general neural network concepts. The word deep comes when people are now talking about deep network. It's just referring to the fact that there are now can be many layers. In my example, there's one layer of hidden layers. In deep networks, there are typically many more layers of hidden layers. And the idea then, it becomes more and more powerful to be able to generate, to engineer more and more features. And so there is no, as far as I know, there's really no very magical technical definition about what deep means. But people look at so they call the credit assignment part as the number of layers the data are transformed. And so for a feed-forward network, the depth will be 1 plus the number of hidden layers, and deep typically means that there are actually more than 2 hidden layers. And so there you have. This is the magic of deep networks. You might find it a little bit of anti-climax. Okay, so that's it. Please.
Can we go back to the?
Sure.
So I'm just have a good understanding but so assuming you have values for demographic.
Sure. Yeah.
I'm assuming that this function G exists sort of within one of those source codes and will lead to what happens if the 2 different pieces of information at one cluster.
Well, if that's how you wire it, that is it. They don't. So if I understand the question, well, is that 2 input nodes, they got wired into 2 different nodes in the hidden layer? Is that what you're asking? And you are asking about how they would be aggregated.
Yeah,
Right. So suppose as a designer that the attitude to product is not going to have any impact on customers in segment 1. Then you might then there's no need to make sure that the input from this node would eventually pass into segment 1. Now, typically, we don't know about a lot of these things, and you will just connect everything. And this is why even in this example, everything is split into every node in the hidden layer, and then everything in the hidden layer is split into segment 1 and segment 2. So there's a very common architecture, but the general answer to your question is that there's a lot of leeway. And that's one of the things that I'm going to warn you about as a user of deep learning is that there's so many degrees of freedom when it comes to how you set up the structure. The results could be drastically different, as you could imagine. And so there is actually one of my slides that now you see a lot of people publishing papers and they like what they're seeing, but actually many of them get the results, but it could be very sensitive to the structure, to the internal wiring of all this length. Yeah.
So how much actually goes into inference of these layers as like an actual unit, or you just kind of basically optimizing error rates by trying to reconfigure like weight and layers, or you actually able to kind of infer any sort of information from like the batteries.
So the more standard there are now all kinds of very advanced tools, but one of the more so I would say that the classical way of neural network learning is to learn all the weights, on the edges. It's not going to change, so it typically doesn't really change the wiring. Now, if you have a wire, so one of the reasons why you connect to everything is because when you connect to everything, well, the weight could be 0. Because when you say that the weight is 0, it basically means that that link does not exist. But if you don't have a link, the weight cannot be there. So this is actually one of the reasons. This is the simple way is to connect every node in the previous layer to every other node in the next layer, because you do allow the weights to be 0. Does that answer your question? Now, there are obviously more advanced research where people try to learn about the structure, and so there's a family of techniques that do that. But I'm just, for the most part, the classical method is the structure, you learn the weight.
Because like in my head, when someone says like a black box, my first thought is like a hidden layer or something like that. It's curious because it's like how much of that information can you actually use to make basically domain specific inference, or is it basically again trying to optimize and like classification?
Well, let me come back to the black box issue a few slides, after this. There's a lot of issues about the interpretation of it. Sometimes it's the weight, sometimes it's the overfitting, and so on. Any other questions? So one of the reasons why deep learning approaches or neural network approaches are very popular is that they have been very successful in many applications outside of biomedical, outside of bioinformatics, and so it's very natural for researchers in our world to say, well, can I do something similar? And in fact, I listed 3 examples here. 3, not just individual examples, but 3 categories of applications. Image recognition. Now there's a field called radiomics, computational radiology. You're trying to take genomics, and then you also take image features, and you try to combine them. You feed into a neural network. Let the network construct the features. And so one example here is Imperial College in the UK. They provide an image-based assessment of traumatic brain injuries. I'm sure maybe once every month or once every 2 weeks you heard from the news that some algorithm is now outperforming human in certain types of cancer or in certain kinds of diagnosis. And so a lot of those examples kind of fall within, a lot of it has to do with imaging in many cases, and because there might be a lot of information that's being captured in the pixels that the human expert cannot really interpret or digest. And then maybe when you have a neural net which has 1,000 nodes, maybe that is able to do a better job. Drug design is another example. Drug design is so there is this field that's called Chem Informatics, and part of it is virtual screening. So in the old days, when pharmaceutical companies want to build new drugs, they need to do a lot of what they call the actual screening. But now you could do it in silico. Now you could do it in silico, and you are just trying to rank. You try to rule out, because the actual screening takes a lot of time, takes a lot of money. So you want to do a simulation of sorts, and so you do a massive amount of screening, but then you build a neural network to try to rank or try to predict the ones that they are not going to work, and so therefore you can ignore them, and then you just keep the most highly promising one, and then you will then feed into the more conventional way. So this is this is done for a long time, and in terms of virtual screening, but I think it's more recent, maybe in the past 5 years, when the virtual screening approaches now is very, very neural net based. So obviously in the clinical domain, there's massive amounts of electronic health record data, clinical data, and an example here is in LA, Children's Hospital use the EHR data to improve pediatric diagnosis of certain conditions. And then closer to the interest of this group is bioinformatics. This is just one example. I'm sure many of you know many other examples in this where deep learning has been applied and applied quite successfully to various bioinformatics problems. Now I'm going to this is the last slide of the first half of my talk, and I'm going to come back to some of the discussion. So we talked about how the basic structures of neural network, what deep learning means, and we talked about how it has been successfully applied. The title of this slide is to say that, well, the fact that there are 5 Nature methods that have been published and is using a deep learning approach, does it mean that everybody should be doing it? Should I be doing it? And this is where this, bias beware, slide. One of the reasons why I want to open the black box and show you the inside of a neural net is to say that inside each unit there are all these links and there's all these weights that you need to estimate when you are trying to learn from the training data. And then imagine as you connect all these different basic units to build different layers, there are a lot of parameters. So the learning exercise that you are doing in machine learning for whatever learning problems that you're trying to solve, you have a huge number of parameters that you need to estimate. So when you hear people say that deep learning methods are data hungry, that's a vast underestimation of what hungry means. So, okay, this is like 7 p.m. I'm sure some of you are getting hungry. But so this is literally like a lot of this method, and particularly depends on the structure of your deep networks. You're really talking about dinosaur kind of hungry, really hungry. So when you are not dealing with a problem where you have a lot of high quality training data and you use a deep learning approach to it, it probably won't work. It probably won't work because you are going to result in very serious overfitting because there are just too many parameters. Let me just give you a simple contrast. In a typical 2-dimensional linear regression, how many parameters do you need to estimate? So you try to fit one straight line. You have the intercept, you have the slope, 2 parameters. That's a linear regression. And you imagine that in a lot of the examples that we do in biomedical research, we probably need 1,000 points to try to estimate 2 parameters. Well, if you have 20,000 parameters, how big would your data set be? So deep learning is great for big guys like Google, like Facebook, huge amount of data. They could do it. It doesn't mean that the little corner store research lab would be able to have access to that kind of data. So this is very, very important. We talked about structure. you know, how you wire them, it's very, it could make the result very, very sensitive to the structure of the underlying layers, how many layers, and how to, you know, whether certain links are deliberately not connected simply because you want to reduce the complexity of the network. It's very important, but a lot of the published papers, actually, they would never, I doubt, as I mentioned earlier, they would not do some kind of, you know, Affiliation or robust analysis as to how the results would change relative to some variation of the structure of the network. In other words, a lot of the results are very, very sensitive to the actual structure of the network. So that does not necessarily, I mean, it doesn't mean that it's necessarily wrong, but it's just not giving you a lot of confidence that the results will be generalizable. Now then we come to the another comment about the black box nature. Interpretation is hard. Now this is why there is now a very active research area in machine learning, which is called interpretable machine learning, particularly interpretable deep learning. They are trying to create deep learning methods, more advanced deep learning methods, where you could provide much better interpretation. In the way that I just, in the simple way that I show you for the basic unit. So these nodes have very typically have no interpretation to them. And then once you do the, even in our example of segmenting the customers into segment 1, 2, 3, you would rarely open up this black box and say, hey, look at the hidden layer. What are the ways? Which one is the most important? You know, sometimes people do, but you know, for the most part, neural network is very good when you are trying to use it to do when you're trying to improve performance. If you are dealing with a performance problem, suppose you will need to classify certain things, as in my segmentation example, and you need to boost the accuracy of your prediction, you need to boost the area under the ROC curve. Neural net is a good tool. But if you are trying to solve a problem where you want to generate insights, or if you're dealing with a problem that the results will never be used unless people understand why I'm getting, you know, better results, then the classical deep networks are not going to be very useful to you because of this point that interpretation is. Any questions or comments? So this is the first half of my talk, and I'm going to show you an example. But I assume that many of you actually come here because you want to know a little bit more or want to talk about deep learning. I open the floor and yeah.
You mentioned that the importance of the high quality data. I wonder what your definition of what high quality is. Because I thought initially you wanted to go with low quality for training to account for variability in classification. What do you think are the criteria for obtaining high quality data?
Well, I think maybe I should have put high quality quote, because I don't think I have any technical definition of what high quality is. Typically, not necessarily in a bioinformatics setting, is that you want a data set that have been verified about its correctness. So let me give an example. In a biomedical case, suppose you are using the patient data. A higher quality training data set to make sure that you don't have a lot of columns, there are a lot of missing values. You have checked that everybody's age is between so I don't know, feed into the sequencing discussion earlier in the previous talk. Maybe if you are using it for training, maybe you want to use data where the error rates are lower rather than the error rates are higher, because then maybe particularly when the data set is not large enough, maybe the neural net actually picks up on a lot of nuance that actually happens to be there. And so high quality is old one. But I think it's just wanting to make sure that just like when, imagine that you are trying to teach a kid to do something. You want to show him or her a lot of good examples, not a lot of bad. He's going to learn the bad thing.
Do you have any suggestions on, I just look like a sort of online course of assumption. Do you have any suggestions for theoretically on the size? Assuming you have the data, how big
No, there's really no heuristic about, I think it's a good thing that I kind of mentioned about good old linear regression. Even if you are using, linear regression, when you are doing a power calculation, you know how complicated it could be, because it also, as you know, in a power calculation, the minimum amount of the data that you need obviously depends on the effect size. And so there's no difference here. So in my example, if segment one of the three segments of the customers, they are highly different from each other, well, the general rule is that maybe I would need a smaller training set. Well, if the profile of the segments of customers are highly overlapping, well, it is a harder problem that I need more data. Now, but I don't think you would, nobody will be asking, you submit a grant, and then you are talking, I'm going to use it now. As far as I know, there is no power calculation when it comes to, now I have 2,500 parameters that I need to estimate, and now what is the minimum data size? So there's no rule of thumb in terms of the size. What I could say is to answer a different version of your question. And the different version of your question is that you get all the data that you get. You get all the high quality data that you think is the best of your ability. You fit into this thing or into any learning method, because that's the best that you can do. And then you need to make sure that you have good practice, like doing cross-validation, doing random sampling. If you take out certain things, you might results change a lot, but the accuracy drops a lot. So you do those standard things to help you, to at least tell you, not so much answering the question of what is the minimum size, but it's answering the question is for the given size that I have, how much can I trust this size? So at least for the best that you could do, how far? So that's a much weaker answer that you could. Yeah. Yeah. Yes. Well, thank you for asking the question. Part of the reason why you connect is that some of these nodes may actually be trying to capture the interactions between them. And so oftentimes one of the contrasting points is that, and it's also, we're not planning about talking about this, but it's a good thing that I mentioned about linear regression. linear regression is a very simple method, as we know, and is only able to handle linear relationships. Neural network, by definition, can do a lot more. So you can handle non-linear relationships as well as highly interactive relationships. So this is what this thing is good for. And yeah, so absolutely. So the point is that you could capture a lot of those things, maybe even non-linear relationships, because your function G may not be linear. And so you could do a lot of things, but because it's so powerful, it also means that you need lots of data to be able to trust this. Yeah. Well, I think that I mean, in a way, I mean, I don't think we have a universal answer about how we could avoid overfitting, except to say that use as much data as you could get, and then you test whether you're overfitting or not. And this is why people still do all kinds of validation, use different different data set, and so there's these are kind of best practice. But the nature of the question is that is there a magic method that people could use that would guarantee that there's no overfitting? Then I don't think there's such a thing. Now, so just the simple rule of thumb is the simpler the model it is, the less the chance of the overfitting. Given the same training set size, if you have a simpler model like linear regression, you are less likely to be overfitting. Versus a neural network that has 2,000 parameters, you are more likely to be overfitting. But at the same time, the simple network may not capture the interaction, may not capture non-linear relationships. So even if you have a model that is not overfitted, maybe the model is less useful. So there is no magic answer to where the sweet spot is. So okay, maybe I'll go to part 2 of my talk, and then we'll come back and actually talk about some of them. So part 2 of my talk is some work that one of my PhD students, Shi Yang, has been doing for the past year or so. And he is very interested in predicting what RNA are going to bind or interact with proteins. And so he's interested in this RPI problem. And so RPI, I think many of you know more than I do. I'm not a biologist. That RPI is all this interaction is everywhere in the cell, so it's a very fundamental problem, trying to understand what RNA are binding to which proteins. It will allow us to understand the biology a lot better, mechanisms, and so on. And the binding, so the proteins, the RNA binding proteins, interact with the RNA sometimes because of the sequence, sometimes because of the structures, sometimes because of both. And this is a very, very busy slide, and the only reason why I have this slide and the next one is not so much about going through the biotech side, because I actually don't fully understand a lot of the details on the biotech side myself, except to say that there are different kinds of methods that generate data about RBPs, about the RNA binding proteins. What proteins bind with what RNA probes. Different ways of doing it. RNA compete, for some of you who might be in the field, is actually one of the most dominant. Well, at least it produced one of the most dominant data sets that is high quality for those proteins and for those probes. And so a lot of these prediction problems are actually based on RNA compete data. So this is in vitro. The next slide is very similar in the sense that there are different techniques for in vivo. And again, there are iClip, and as I'm saying, I won't be surprised if many of you know a lot more about these technologies than I do. But there are all these different data sets. And what is interesting, so that may feed into the earlier question about what is higher quality, what is lower quality, is also the technology and which technology, and so on. Now, the prediction problem that we are trying to solve actually have two shades to it. There are two subproblems. The first problem, people call the partner prediction problem, is to predict whether protein A and RNA B interact at all. So this is, do they interact? Whether they interact. This is partnership. The interface prediction problem is, well, where do they bind? So if they interact, where? And in fact, some of the data, you could even argue that embedded in the interface prediction problem is just saying, how strong that they bind. Whereas the first problem is almost just like a binary answer. Do they bind or not? Whereas the second is where and maybe how strong. So this is a bit, given the importance of understanding both the interface problem and the partner and the partner problem. I'm just showing you in one slide. This is not intended, to go into detail, except to say that some of the approach. This is overview of the literature in this general area. And on the upper half is protein centric, so it starts off from protein, trying to understand what RNA would bind to certain protein. The bottom half is RNA centric. It's like the reverse. You have certain RNA in mind, and you want to understand which proteins are binding to them. And so there are various methods that have been proposed about whether it's interface prediction or whether it's partner prediction. There are papers that just focus on using sequence features. There are papers that add in other features, and there are papers that sometimes they don't use sequence features. For example, they may just use structure. And so you have different structures, and then on the RNA side, you might have motif discovery as features, and so there is different methods that have been proposed, I would say, in the past 10 years. So this is a very general overview of the field. Now, what we do and what we're going to spend maybe 10 minutes talking a little bit about our work is one of the paper in here. And so this is the paper that was published in Nature Biotech about 4 years ago, 2015. It's called Affinity Regression. And so the full title of the paper is trying to predict the recognition of nucleic acid binding protein, RBP. They are trying to do both the partner and the interface problem. So they were using in vitro data RNA compete. The reason why I want to talk about this is because it actually is an interesting paradigm. So what I'm showing you in this diagram is, this is a typical Netflix. So think about Netflix. These are people who are users, these are movies, and so this is a typical recommendation system problem. People have seen the movies and they give a rating. So a 5-star rating is I love this movie, or I like this movie with a 4 star. Okay. the other hand, I hate it. So you have a matrix here, the user, the movie, and then you have the affinity. Okay, how much you like. So this is a typical Netflix recommendation system, paradigm. And one of the interesting things about this particular paradigm is so-called the co-start problem. When you have a new user, and you know very little about the user, you are going to what are you going to recommend to the new user? And so that is called co-start problem. Now, the reason why all of a sudden I talk about movies is, well, affinity regression is exactly using the same paradigm. Imagine that the items or the movies are the RNA probes, and then the users are your proteins, and you have a rating matrix that gives the intensity of for each pair of protein, each pair of RNA probe. You already have that intensity. They might be 0, which is like hate, or they may be really, really strong, which is like love, as in the 5 out of 5. And then you have all these different features depending on the protein and the probe. And you essentially is dealing with the problem is when I have a new protein, how could and I want to predict which probes are going to have a strong enough intensity binding to that protein, how do I make that prediction? And so it's exactly like the Netflix recommendation problem. And so this is what they're trying to do. And the affinity regression problem, they have a few tricks to allow them to be able to apply that in this context. So in our work, probe grading is the work that Xu Yan has done. It actually extends affinity regression in 2 ways. So I just have one slide that explains to you what he did or what we do. And here I'm showing you a neural net construction. We actually started off using now a very popular technique in the deep learning world, which is called embedding. And the idea of an embedding is really just trying to condense a lot of contextual information. So imagine that we have RNA sequence here on one side with protein. With this is the sequence. And then we use an embedding to capture the essence, the contextual information about the RNA sequence and the protein sequence. And so that is a embedding approach. This is a method. This kind of approach has been proven to be very popular, very successful in image compression as well as in natural language processing. So once we have that, once we capture the similarity, because the idea is that if I have a new protein that I know very little about what it does, the first thing I want to do is somehow I want to find all its similar proteins. And based on all the similar proteins, then I'm going to feed that into a feed-forward neural net. This is not even the recurring neural network that we're using. We just go one way, and then it's going to predict the final intensity. So we're using some deep learning techniques, but the layers is really actually very, very shallow. So this is just to show some of the details. We were talking about numbers. So the similarity part doesn't need to be our own data. So this is why we could use a lot of open data to do that. And so this is very large number, up into the 500,000, and it allows us to just really capture that similarity between proteins and RNA. And then the part where we are then doing the feed forward, we are using RNA compute data, 2 protein families that we work on, the RRM, which is 164 proteins, and the homeo family, which is 218 proteins. Then the nucleic acid part is well into the hundreds of thousands when it comes to the number of RNA probes. This is one or two very simple result slides. All we're trying to show you is that every grade curve is some kind of a baseline that we are comparing. And the whole rating is predicting for each protein. So this is actually sorted in ascending order of accuracy. And so this is why you see a kind of ascending curve. And as you can see, in every case, what rating has been able to do is actually quite a bit better than the baseline. Now, but the most important thing is to compare with affinity regression, because this is how we want to build on top of the work. So the red line is affinity regression prediction, and as you can see, for the most part, so the top one is for the R family, and the second one is for the homeo. You can see the most, so the green curve is almost, much more often will be above the red line than below the red line. In fact, what we want to show, particularly at the beginning, is when the proteins are really hard to predict. In fact, what we are doing is a lot more successful. When the proteins are actually easier to predict, we don't necessarily gain too much, even though we are not really paying a big price for it. So the most important thing for us is that it's some part of the x-axis, because when we really have very little information about those proteins, we are actually doing significantly, or there is statistical significance in the performance. So I am done. Right, 7:15, and so I want, so the first half of my talk is just to give you a little introduction for those who might not know what deep networks are, and you are curious what it is, and I hope that I have opened the hoods, for you, that at least now you could think that, well, actually deep network is not anything different from the old conventional neural network, except that it's a lot more layers. And because of that, I think now you need to remember that it really is very, very hungry for training data, dinosaur hungry for training data. And the deeper the network, the more hungry. There's a bigger and bigger dinosaurs, and so you really have to use it. Knowing that if you're going to use this big hammer, you better have enough food, enough feed, enough to feed the network. I show you in the last 20 minutes very, very quickly, for those of you who might not know, the RPI prediction problem, RNA binding the protein, and just showing you in our work, I think it's actually been quite successful in how we set up the network. But even in that, the layers are extremely simple. And in fact, we deliberately set it up. We don't want to do things blindly. We actually set things up in a very intentional way. And that's why we don't want additional layers. We just want to make sure that we capture certain things, and so the networks, the feed-forward network that we use is, by today's standard, it's actually quite simple. And so with that, I thank you for your attention.
Thank you, Dr. Ng, for that talk. Thanks, and also thanks for helping us to think about deep learning and machine learning in general, because it's a very popular topic, but I think it's important for us to know how to best use these tools. So I think we'll save questions for just during the networking session. You can feel free to ask Dr. Ng any more questions you might have. So I think we have pizza and drinks here, so please help yourselves to those. Thank you. Oh, drinks will be around the corner. So, and drinks is by donation for anyone who wants drinks. Thank you.
I didn't know that. So I
I see.